In this video, I'm going to be breaking down how exactly B and T cells work using this very simple to understand flowchart. Our adaptive immune response is a part of our third line of defense. So this is where the response becomes very specific to each pathogen. And how this is triggered is because of these molecules called antigens. Antigens are these unique marker molecules that are found on the surface of cells. Think of antigens as these name tags that are found on pathogens, and they signal to the body that this is a foreign substance and we need to initiate an immune response. Our own cells also have antigens, and we call them self-antigens. And these are like your ID cards. Our immune system knows our own self-antigens, and it will only attack cells that have non-self antigens, which can be found on things like pathogens, so bacteria and viruses, or other foreign materials. So now that we understand how the adaptive immune response gets triggered, let's have a look at the main players in this response. The main immune cells involved will be your B and T cells. These are what you call lymphocytes, which are a type of white blood cell, and they each have a different job. Once the body detects an antigen, these B and T cells will undergo a process of differentiation, where they'll multiply and specialize into different types of cells. Let's start with B cells. B cells are a part of what we call the humoral response. So they help mainly to fight off pathogens that are found outside of cells. So they could be found in the blood or lymph, for example. And their main path of attack are through these molecules called antibodies. Antibodies are these Y-shaped proteins that bind to a specific antigen that's found on the surface of a pathogen. And they help to neutralize that pathogen as a result of it. And the main cells that produce these antibodies are called your plasma cells. When the body activates the B cells, most of your B cells will differentiate into these plasma cells, which produce a lot of antibodies that bind to the antigen that's found on the pathogen and help to deactivate it. At the end of it, all phagocytes will go and engulf the cellular debris that's left over. Now, alongside the plasma cells, the body also creates these memory B cells. So these cells remain dormant in your body for even years after an infection. And what they do is that they remember the antigen that is on that pathogen. And if you get infected again by the same pathogen, your body will be able to quickly recognize it and initiate a much quicker and swifter response. So in the case of B cells, what happens is that these memory cells will rapidly divide into lots and lots of plasma cells. And as a result, they will create a larger concentration of antibodies in a much shorter period of time. In summary, there's two types of B cells. You have your plasma cells and memory B cells. Plasma cells are the ones that produce the antibodies. Memory B cells stay in the body even after the infection has been fought off, and they will be the ones that quickly differentiate into your plasma cells again and create even more antibodies at re-exposure. So memory cells are the key things that actually help with long-term immunity. And these play a key role in vaccinations, which we'll learn about next time. T cells are part of the cell-mediated response. T cells do not make any antibodies. They directly themselves help destroy the infected cells. So the main killer cells in this will be your cytotoxic T cells. So the cytotoxic T cells, they have these receptors that will bind to that self-antigen on an infected cell. And once it does that, it's going to release these chemicals called cytokines. And cytokines will destroy that infected cell. Once all these infected cells have been destroyed, then phagocytes will go and engulf them. Alongside these cells, you again have your memory T cells. So memory T cells, just like memory B cells, they're going to remember that antigen. And if you get exposed to the same antigen again, then the T cells will quickly differentiate into your different types, and they will carry out a much quicker and swifter response upon your re-exposure. There are other types of T cells involved, which we'll look into in the next section. Now that we understand how B and T cells work, we also need to look at how these actually get activated to carry out their responses. So here's the thing, B and T cells don't just jump out to destroy a pathogen as soon as it's entered a body. So there's a whole process involved in activating them. Let's have a look at one of the main ways that B and T cells get activated. So we're going to start with looking at antigen presenting cells. In this scenario, we're looking at a macrophage, which is a type of phagocyte, and phagocytes typically will engulf a pathogen. The first step in this process is that macrophages that are circulating around, it could come across a pathogen. And what it's going to do is that it's going to engulf that pathogen, and it's going to present the antigen of that pathogen on its own cell surface. So the point of it doing that is to alarm and to signal the body that we need to carry out an immune response. And how it does that is through these things called helper T cells. So in general, helper T cells play a role in activating the immune response. They will bind to that antigen that is presented on that macrophage and release these chemicals called cytokines. So cytokines are these signaling molecules, and they will tell the B and the T cells to begin differentiation. So what happens is that inactive B cells, they will then quickly differentiate into your plasma cells or your memory cells. While on the other hand, the cytokines are also going to trigger T cells to produce more cytotoxic T cells to kill that infected cell, as well as some memory T cells. So there is another way that B cells can get activated. And this usually involves a special protein found on the surface of these inactive B cells called B cell receptors. Each receptor will bind to a specific antigen. So once this inactive B cell has formed this complex, it's going to process that antigen. So it's going to break it down and present a piece of it on its own self-antigen, which we can call a MHC class II molecule. If this helper T cell then recognizes this antigen being presented, it will go ahead and bind to it. And again, this will trigger the release of cytokines, which will give an inactive B cell the signal to begin activation. This will allow it to fully differentiate into your plasma cells and memory B cells. So my goal with these videos is to help break down biology concepts in a way that's really simple to understand and doesn't overcomplicate it like some textbooks can do. If you found this video helpful, then please give it a like and check out my other HSC biology breakdowns. And if you are looking for more structured support, I also offer small group Year 11 and 12 tutoring, and you can find more information on my lessons in the description down below.